Hello everyone. This chapter we are going to talk about how estimating chlorophyll A concentration using remote sensing technique by group 2. My name is Sonya Theresia with student of number 1913511047. Before we jump to remote sensing technique, we have to know why we estimating chlorophyll A. Over the years, increasing population and urbanization has stressed the circumstances. Monitoring of chlorophyll A level is the way to estimate the algal growth in water. Because chlorophyll A is one of the ecological indicator, so it can be used to evaluate the ecological status of marine environment. Basically, this pigment cell is used to energy from sunlight to produce oxygen for life sustaining. To monitor and measuring chlorophyll level is a way of tracking the algal growth. And we can use remote sensing technique to estimation and creation of a map of chlorophyll A distribution level. Nowadays, several algorithms have applied and developed to chlorophyll A map in marine area, estuary, and freshwater mass. So, the aim of the study is to be a source information on how to use remote sensing technique in marine fields.
Hello everyone, my name is Gusti Galih, Ksatria Mahadhika Nurdyputra, and in this chapter, we're going to talk about basic theory of remote sensing. And here's the topic of today, electromagnetic spectrum, passive sensor, and active sensor. The electromagnetic spectrum ranges from the shorter wavelengths, includes gamma and X-rays, to the longer wavelengths, includes microwaves and broadcast radio waves. There are several regions of the electromagnetic spectrum which are useful for remote sensing. For most purposes, the ultraviolet or UV portion of the spectrum has the shortest wavelengths, which are practical for remote sensing. This radiation is beyond the violet portion of the invisible wavelength, hence its name. Some Earth surface materials, primarily rocks and minerals, fluoresce or emits visible light when illuminated by UV radiation. The light, which our eyes, our remote sensors, can detect is part of the visible spectrum. It is important to recognize how small the visible portion is relative to the rest of the spectrum. There is a lot of radiation around us, which is invisible to our eyes, but can be detected by other remote sensing instruments and used to our advantage. The visible wavelengths, as we can see in the right image, cover a range from approximately 0.4 to 0.7 micrometers. The longest visible wavelength is red and the shortest is violet. Common wavelengths of the we perceive as particular colors from the visible portion of the spectrum are listed below. It is important to note that this is the only portion of the spectrum we can associate with the concepts of color inside our eyes. The next portion of the spectrum of interest is the infrared, or IR. The infrared region, which covers the wavelength range from approximately 0.7 micrometers to 100 micrometers until more than 100 times as wide as the visible portion. The infrared region can be divided into two categories based on their radiation properties: the reflected ER and the emitted or thermal ER. Radiation is in this reflected ER region is used for remote sensing purpose in ways very similar to radiation in the visible portion. The reflected infrared covers wavelengths from approximately 0.7 micrometers to 3.0 micrometers. The thermal infrared region is quite different than the visible and reflected infrared portions, as the energy is essentially the radiation that is emitted from the Earth's surface in the form of heat. The thermal infrared covers wavelength from approximately 3.0 micrometers to 100 micrometers. The portion of the spectrum of more recent interest to remote sensing is the microwave region from about 1 millimeters to 1 meters. This covers the longest wavelengths used for remote sensing. The shorter wavelengths have properties similar to the thermal infrared region, while the longer wavelengths approach the wavelength used for radio broadcast. Remote sensing systems, which measures energy that is naturally available, are called passive sensors. Passive sensors can only be used to detect energy when the naturally occurring energy is available. For all reflected energy, this can only take place during the time when the sun is illuminating the earth. There is no reflected energy available from the sun at night. Energy that is naturally emitted, such as thermal infrared, can be detected day or night, as long as the amount of the energy is large enough to be recorded. Active sensor, on the other hand, provides their own energy source for illumination. The sensor emits radiation which is directed towards the target to be investigated. The radiation reflects from the target that is detected and measured by the sensor. Advantage for active sensors include the ability to obtain measurement anytime, regardless of the time of the day or the season. Active sensors can be used for examining wavelengths that are not sufficiently provided by the sun, such as microwaves, or to be the better control the way the target is illuminated. However, active systems require the generation of a fairly large amount of energy to adequately illuminate target. Some examples of active sensors are lasers, fluorosensor, and a synthetic aperture radar or SAR.
The next section will be introduced by me. My name is Miguel Jeremy Amadeus Tamaela with student number 19135111076. Now we are going to talk about what is exactly chlorophyll A. Chlorophyll A is one of the most important photosynthetic pigments for aquatic organisms. There are other types of chlorophyll known to date that are owned by phytoplankton. To name some of them, chlorophyll A, chlorophyll B, chlorophyll C, and chlorophyll D. Out of all these pigments, chlorophyll A is the pigment most commonly found in phytoplankton. Therefore, the concentration of phytoplankton is often expressed in terms of chlorophyll A concentrations. Indicators of water fertility can be measured from the content of chlorophyll A. Water fertility is influenced by several parameters. For example, sea surface temperature, otherwise known as SST, wind speed, and upwelling. One of the functions of mapping chlorophyll A location is that fertile water areas or areas where upwelling occurs are often marked by fish catching activities. One example of fishing area in Indonesia is the Fishery Management Area, otherwise known as WPP.
Hello, my name is Trisna Dewi. Student number is 19135111023. Here I will continue to explain the material about remote sensing used in observing of the chlorophyll A in the oceans. Okay, next we are going to explain chlorophyll A. The measurement of the chlorophyll A is based on the value of the reflections or reflectance of particles containing green substance on the surface of the water and that are captured by remote sensing satellite sensors. The higher green matter value obtained the sensor of chlorophyll A is which one? A sensor is Sea-Waves is satellite burner sensor designed to collect biological data of the global oceans, and its main mission is to measure the chlorophyll produced by marine phytoplankton. Okay, next we are moved to the science of launch of the satellite carrying the Sea-Waves sensor in 1997. Data on phytoplankton chlorophyll concentration have been available, which can be used as basic data for management of biological research in Indonesian water.
And now to introduce myself, my name is Ahmad Ramadhan and my student ID number is 20135111031. And my job is interaction between color A, observation, and satellite. Next. Okay, basic interpretation of multispectral image. Sensor catches in digital form and are sent to Earth for further processing. Multispectral image interpretation requires several stages of workflow, including correction, calibration, and so on, according to the required analysis. Next, simple processing of multispectral image. For this discussion, we will try to do the band composite or band composite RGB technique. This technique is done by combining a maximum of three different bands in the RGB, red, green, and blue color spectrum. As we know, each band records light over a certain range of wavelengths. Each of these bands can be interpreted in black and white image. Black means no or little light is called. Brighter white means that the object reflects more light wave at that wavelength. Next. And figure 3 is simple illustration of composite RGB processing of a multispectral image. For more details, see the illustration in figure 3. For example, we choose band X, Y, and X, Y, and Z, each of which capture like a different wavelength. In this simple example, area has three objects where each object reflects like a different wavelength. If each of these bands is mapped to a certain RGB color and then combined, a composite image will be produced according to the characteristic of the band and the corresponding RGB color. If an object reflects light in more than one wavelength range, the resulting composite color will also differ according to which color is dominant. As a simple example, chlorophyll leaf, when viewed with the A, will be green, dark green, or light green, depending on the range reflect light wave. But did you know that plants with chlorophyll also reflect most of the infrared wave? Next.
Allow me to introduce myself. My name is Gede Bhujangga with student number 2013511054. Here I will explain a little about the stage of image data processing from chlorophyll A observation. As seen here are two maps of different chlorophyll A observation that have been processed. But how is the process being done? The first step in the stage of processing satellite imagery is done by collecting image data, where the download of the image data can be done through websites such as NASA Ocean Color website. The second stage is called geometric correction or koreksi geometric, where the purpose of this geometric correction is done to reduce the geometric distortion of the Earth's surface object in the imagery caused by the curvature of the Earth's surface and several other factors such as variation in satellite height, satellite firmness, and satellite speed that result in the spatial position of an area in the image in accordance with actual position in the Earth. The third step is called cropping or pemotongan citra, where this step is done with the aim of narrowing the area of existing data and focusing the data on the research location you want to get. The fourth step is called spatial subsetting. This step is done to cut the image area that is not needed in this study, where the data can then be separated or even eliminated to adjust the research location to be observed. The fifth stage is called cloud masking or pemisahan awan. Cloud separation is done using an algorithm as follows. The sixth step is the calculation of chlorophyll A concentration using algorithm, where the algorithm adjusts to the satellite used to get the data. In this section, the algorithm for calculating chlorophyll A concentration is used when the data obtained come from MODIS Aqua and Envisat Maris satellite. The seventh stage is called ground truth. Ground truth itself is done by taking sample at the research location that wants to be obtained and carry out calculation in a laboratory. For the calculation of chlorophyll A concentration, there are formula described as follows. The last stage is called validation or validasi. This stage have the goal to be able to further analyze the correlation between in situ data and image processing result.
So this is one example map of chlorophyll A distribution as the result estimating chlorophyll A using remote sensing technique. As you can see in this two-figure map distribution of chlorophyll A, I used case study from paper by Seima, Merv Kaimas, and Arsen Ates. They used Marmaris for the area of interest. Marmaris is a coastal city located in the district of Mungla, southern west of Turkey. In this study, they used Sentinel-2A with two satellite images, that is May 2017 and May 2018, were taken from the Marmaris Bay. Sentinel-2A images are used as a representation of the distribution of chlorophyll A. The processing of Sentinel-2A imagery are done by using ArcGIS 10.5 software for radiometric correction, calibration, cropping, and implementation of algorithm. The chlorophyll A value in the study area have significant difference. We can see in May 2018, the range of chlorophyll A amounts were higher than May 2017. The increasing of chlorophyll A value is evaluated an indicator of organic pollution in the marine system. Chlorophyll A level is high in the area near wet coastal zone. It might be the reason of production of phytoplankton, be the consequence of the anti-additional nutrient supply by runoff the stream flow. The value of chlorophyll concentration in the study area, according to Sentinel-2A imagery processing, that the range value of May 2017 were estimated between 0.01 to 5.5 microgram per liter, and the value of May 2018 were estimated between 0.028 to 8 microgram per liter. So that's it from me. Thank you.